Okay, so glycolysis in the citric acid cycle, the main purpose of those was to make these electron carriers NADH and FADH2. These molecules, they represent potential energy. Potential energy in the form of stored electrons. So we would take NAD plus, and then we would add electrons to it. We would get those electrons from glucose, or some derivative of glucose. That's where the electrons came from. And then by adding the electrons in the form of a hydrogen bond, we will then create NADH. Then the electron transport chain, the idea of that is we are then going to remove those stored electrons from NADH and FADH2. In other words, we're going to oxidize NADH and FADH2 and use the energy from oxidizing those two electron carriers to pump hydrogen ions to the inner membrane space. You got to know the locations of these things. The inner membrane space, that's where the hydrogen ions are at. And in these hydrogen ions, what will happen because of that pumping, you'll now have a higher concentration of H plus in the inner membrane space and a lower concentration of H plus in the mitochondrial matrix. That creates an energy gradient where these hydrogen ions, they want to go from more to less. But there's only one option for them to get back into the matrix, and that is through this protein complex called ATP synthase. So think of like a wind turbine. This is literally a turbine, world's smallest turbine right here. The hydrogen ions would be like the wind here, and Kylie, it's even days.
Oh, sorry.
Here, probably it's done. These hydrogen ions are going to go from more to less, and the energy of them going down their gradient is going to attach a phosphate group to ADP, thus generating ATP. Really cool process. Really rewarding to make sure you understand that. So then next slide, kind of showing that ATP synthase in more detail. So here's each of these gray balls that represents the hydrogen ions. So you have that hydrogen ion gradient, and then these hydrogen ions moving from the inner membrane space down through, so this is the ATP synthase in a little more detail. You don't need to know like the parts of it so much, but understand the idea here. These hydrogen ions going down, it's gonna spin a bunch of different things. It's gonna spin a rotor, which spins an internal rod, which finally turns a catalytic knob. So the catalytic knob, that's what's going to do the catalysis. It's going to catalyze the reaction of ADP plus inorganic phosphate to ATP. Okay? And there you go. Any questions about ATP synthase? You should be able to, maybe not in like this level of detail, but in words describe that general process. Okay? All right. And then here's a really good just overview slide of cellular respiration. Remember, with cellular respiration, it's really about knowing what the inputs, the outputs, and the locations of all these things. Meaning, you don't really need to know like the actual like chemical reactions of glycolysis or the actual chemical reactions of the citric acid cycle. Understand that these are oxidation reactions where we're stripping those electrons away from glucose or some derivative of glucose like pyruvate or acetyl-CoA, and attaching those electrons to NADH and FADH2. Then you send those into the mitochondria to do the electron transport chain. And chemiosmosis, I didn't say that word, that's the process of the hydrogen ions going down ATP synthase. Okay? Chemi referring to the chemical H+. Osmosis, think of that's when water's going from more to less. Here it's the hydrogen ions going from more to less. Yeah, Thomas? Oh, yeah, good question. Yeah, the H plus is like where they're originating from. Yeah, it would be coming from when you're removing those electrons. Like NADH has that. This H should be like what's getting pumped out of there. Yeah. The exact mechanism of how exactly that's happening isn't really as important for you right now. It would have something to do with like as these proteins are absorbing the energy from that redox reaction, that causes a change in the shape of these proteins that will cause the hydrogen ions to get sent out. So anyways, okay, going back to this summary slide, when you combine that whole first slide, the electron transport chain plus chemiosmosis, the H plus going through the ATP synthase, those two processes are called oxidative phosphorylation. Oxidative, you're doing you're oxidizing the NADH, the NADH and the FEDH2, and then phosphorylation, you're using the ATP synthase to make all that ATP. It's about 26 or 28 ATP. Okay? And then through substrate level phosphorylation, we can make small amounts of ATP in glycolysis in the citric acid cycle. Okay? All right. Moving on to 7.5. So basically, this is getting into like, all right, well, what happens when you're trying to do cellular respiration and you don't have oxygen? Because if I go back to this picture of the electron transport chain, oxygen is your final electron acceptor here. So that's why you need to breathe in oxygen. So if you don't have enough oxygen, well, then that's going to present a problem because you can't do oxidative phosphorylation. You are only you're limited to just doing the substrate level phosphorylation. When I say that, I mean this. You can only make the 2 ATP from glycolysis. Okay, and that's why that's bad is because 2 ATP is different from like the 26 to 28 that you make in oxidative phosphorylation. So a lot less efficient. Okay. Anaerobic respiration and fermentation. Anaerobic respiration includes the electron transport chain. Whereas fermentation is only going to involve, you only can do glycolysis. Where am I at here? Glycolysis only. Okay, both don't involve oxygen, but fermentation, you're just doing glycolysis. Yeah, did you?
So if you have anaerobic, it's both electron transport and glycolysis?
If you're doing anaerobic, you will do glycolysis, citric acid cycle, electron transport chain. What happens is in the electron transport chain, sulfide, the anion of sulfur, that's the ending molecule for accepting electrons, not oxygen. So know that. So in normal electron transport chain, all of our mitochondria do, oxygen's the ending acceptor, but there are some bacteria that like live in like really oxygen poor environments where they'll use sulfide. Sulfide, that's the anion sulfur, so it's S with a -2 charge. That makes it a really good acceptor of electrons. Anyways, so you got that. Okay, a lot of this slide I already said. Oh, I'm sorry, I said that wrong. It's sulfate, not sulfide. Sulfate is the ending acceptor. Doesn't matter as much. Just know it's some molecule that involves sulfur. Okay. Yeah. So the idea here is like, if you're like a bacteria, or even if like some of our cells, just because you don't have oxygen present, you still want to have some way of being able to make energy. In other words, like you don't want to just like curl over and die just because you can't get enough oxygen to your cells. That's the main idea of fermentation. And well, we just can do fermentation, but like if you're like some bacteria, you can do like the actual anaerobic respiration. All right. So the idea here is, remember, cellular respiration was all about those electron carriers. You still need the electron carriers in fermentation. Okay. So when you do glycolysis, so you still do glycolysis and fermentation, you will make the NADH, right? And if it's normal cellular respiration, you have oxygen present, that NADH would travel the electron transport chain. But if you don't have oxygen, you can't have the electron transport chain functioning. During glycolysis, you will make 2 ATP. So if you just do glycolysis, you make a small amount of ATP. But the issue is you have to find a way to regenerate your NAD+. Because eventually, if you make NADH and then you don't keep regenerating NAD+, you won't be able to keep making the 2 ATP from glycolysis. So you have to regenerate the NAD+ eventually. That's really where the reaction to fermentation happens. And I'll show you the actual reaction on the next slide. All right. Within fermentation, there's alcohol fermentation and lactic acid fermentation. I'll show you the two reactions. So showing you alcohol fermentation. You don't need to know it to like the level of like drawing the chemical structures, but you do need to know the molecules made. So know that in fermentation, you take glucose, you do glycolysis, where you make 2 ATP, and remember that's by substrate level phosphorylation. Okay? And you make 2 pyruvate. If oxygen's present, those 2 pyruvate would then go into pyruvate oxidation to make the acetyl-CoA. Acetyl-CoA goes into the citric acid cycle, and then we go to the electron transport chain, and you do that whole process. But if you don't have oxygen, the pyruvate will then be turned into acetaldehyde and ethanol. The reason why we do this reaction of pyruvate to acetaldehyde to ethanol is to regenerate NAD+. Because as I said a couple slides ago, if you want to make the 2 ATP, but as a result, you'll make the 2 NADH. If you don't ever regenerate the NAD+, you won't be able to continue making your 2 ATP. So that's the purpose of taking pyruvate. And know this, you take pyruvate, a 3-carbon sugar, and you turn into acetaldehyde, a 2-carbon sugar. And then you're going to do in order to make NADH go back to NAD+, you're going to oxidize this molecule. You're doing an oxidation reaction. So you're oxidizing NADH to NAD+. What that looks like chemically, this is a good theme to understand. If you take a carbon to oxygen double bond and turn it into a C to OH bond, or take a double bond to oxygen, make it an OH, that represents adding hydrogens. Like when you add a hydrogen to NAD+, adding hydrogens represents a reduction. Okay? No, that's a good theme. Don't need to know the actual structures quite yet, but understand adding hydrogens is the gain of electrons. That's the gain of, another way of saying that is this molecule is gaining potential energy. This molecule then, NAD+, has lower potential energy compared to NADH. Okay? And then remember, the reason why there's a 2 here and a 2 there is because we do this times 2. Glucose was a 6-carbon sugar, and you made 2 3-carbon pyruvates. So know how to count the carbons with these molecules. No acetaldehyde is a 2-carbon sugar. Acetaldehyde is similar to acetyl-CoA. Acetyl-CoA is what, when you did pyruvate oxidation, that's what you made, and then acetyl-CoA goes into the citric acid cycle. Acetyl-CoA was also a 2-carbon sugar, just like acetaldehyde. That acetyl is referring to a 2-carbon sugar. Okay. All right, questions here? This is how alcohol is made. Ethanol. The all there, the OL, that's referring to that alcohol group, the OH. It's an alcohol group, if you remember from chapter 2. So this is really like, the carbon dioxide, that's why beer's fizzy. What happened to my laser pointer? That's why beer gets kind of its carbonation. So anyways, this is the basic process of how like any alcohol is made. So this is not what we do. Our cells don't do this. This would be like what a lot of bacteria and what yeast will do. Okay? Let me get this over. So I should have said everything here. Like baker's yeast. Oh, that's CO2 produced. Like when you remove the CO2 from what is happening with this cursor here? Give me a sec. That CO2 produced, that's what's going to cause like bread to rise. Okay. That's what's going to cause bread to rise if you're baking something, or again like the carbonation in beer and stuff like that. Okay, now lactic acid fermentation. So this is what our cells will do if we don't have oxygen. So lactic acid fermentation, it's a fairly similar process to fermentation, but notice how you're taking pyruvate and you only do one step. You're not doing 2 steps like in alcohol fermentation. Here, you're still making pyruvate, right? You're still going to do glycolysis to make your 2 ATP by the substrate level phosphorylation. And then we have to regenerate the NAD+, otherwise we will stop doing glycolysis. But here we take pyruvate, and all we have to do is just do a one-step reaction where we're going to take this carbon to oxygen double bond, and we're going to reduce it right there. In other words, we're going to take the hydrogen from NADH, we're going to oxidize this and attach that hydrogen to pyruvate, making it turn into, that would then be called lactate. Okay? Lactic acid, the name lactic acid, is referring to if I were to add another hydrogen here, that would then be the lactic acid. But if you remove the hydrogen, if you're missing the hydrogen, it's called lactate. Lactate is the ionized form of lactic acid. So you've probably heard of lactic acid when it comes to like exercising. So if you're like, especially like runners and stuff, you'll hear like a common wisdom is like, oh, when your muscles are sore, it's because of lactic acid buildup. Interestingly, your book was saying that isn't really like the actual accepted truth now. Like what causes muscles to be sore, the thought now is that it's actually just like micro tears in a lot of your muscles. Because you can make your cells will have cells that will make lactic acid normally, because lactic acid, we could actually use this molecule lactate to build some other things in the cell, and your kidneys and your liver can convert lactate back into glucose. So because this molecule, because you have that hydrogen atom attached there, this molecule still has potential energy, a decent amount. So we can reuse this back into glucose. But it isn't the preferred, this isn't preferred, right? You want to have oxygen so you can do the full gambit of cellular respiration. All right, we got questions here? Good. Don't need to memorize the structures, but no, lactic acid fermentation, you do glycolysis, but then you do in one step turn pyruvate to lactate. And the twos are for what I said earlier. You're doing that process times 2. Because you make 2 pyruvates when you do glycolysis. Okay. This is a great slide to kind of help you get that overall picture of what's happening. So you have glucose. Glucose comes in the cell. Glucose will go through glycolysis. No matter if oxygen is present or not present, say that again, no matter if oxygen is present or not present, you do glycolysis and you make pyruvate. Now, normally our cells are going to you're going to breathe in oxygen, use that oxygen to do aerobic cellular respiration. Aerobic cellular respiration is what we have talked about before this class, where we put pyruvate in the mitochondria. You make acetyl-CoA that was pyruvate oxidation, that process of pyruvate to acetyl-CoA. And then you put acetyl-CoA in the citric acid cycle. The purpose of both of these processes is they're going to make the electron carriers, the NADH and the FADH2, and then these will go to the electron transport chain where we make that hydrogen ion gradient, and then oxidative phosphorylation, that's when the ion gradient is used to then make ATP going through the ATP synthase. Okay, but then if you don't have oxygen present, for us it's usually if like you're working out or like, you know, I don't know, being suffocated, but typically not being, you're just, you know, be like working out or something. Your cells still need to make energy, but then you'll do it through lactic acid fermentation, lactate. Ethanol, that would be the alcohol fermentation. By other I don't really know, honestly, what thinking about other products. Not as important, but no, ethanol, alcohol fermentation, lactate, lactic acid fermentation. Okay? And then where this whole process of no oxygen present, that's happening in the cytoplasm, in the cytosol. Are we good here? Okay. Now, lactic acid, it's actually a pretty diverse molecule. So that's what makes like cheeses and yogurts. It kind of gives it that tangy taste. That the taste of that is lactic acid. And here's something that's chocolate. Like, you know what? Like, you need to make chocolate. You need lactic acid. So the yeast and bacteria that are in the cacao beans. Is that how you say it? Cacao beans. Cacao beans. Yeah, where you make cocoa and stuff. That's dependent on making that lactic acid. Pretty interesting. So, yeah. And there you go. All right, so then you need to be able to compare and contrast fermentation, anaerobic respiration, and then aerobic respiration. So all three processes, you're using glycolysis where you can make 2 ATP. If oxygen is present, then you can go further and do the electron transport chain and make even more ATP. All three are going to be using NAD plus as the oxidizing agent. Remember, the oxidizing agent is the thing that accepts electrons. So there, the NAD plus goes to NADH. And then all three will make, that's how you make the 2 ATP is by substrate level phosphorylation. Okay. Now, how we oxidize NADH, in other words, how you regenerate. So how you regenerate the NAD plus, generate NAD plus, that's going to differ in all three. So if it's anaerobic respiration, you can still do the electron transport chain, but there you're just going to have sulfate as that final electron acceptor. In cellular respiration, it's oxygen doing the accepting of electrons. If it is fermentation, that's when you're going to not do, you can't do any electron transport chain, and that's why you can only make 2 ATP per glucose molecule. So you'll have to just regenerate the NAD plus by making the ethanol or making the lactic acid. All right, getting into like some organisms that will do these processes. Obligate anaerobes, those are organisms that only do fermentation or only anaerobic respiration. In other words, they literally can't survive in the presence of oxygen. If you're obligated to do something, you're like kind of forced to do something, like you have to do it. Obligate anaerobes, they are obligated to live in the absence of oxygen. They're obligated to do anaerobic respiration. Okay? Whereas facultative means like discretionary or optional. You don't always have to do it. So facultative anaerobes, they can do both, right? If they have oxygen present, they'll do cellular respiration. But if they, if oxygen's absent, then they can do fermentation. Bless you. Okay? So we would be facultative anaerobes, right? Like we can do both. All right, almost done with 7.5. A little bit of like evolution stuff. So glycolysis happens without oxygen. So the idea would be glycolysis would be the very first kind of energy system that developed. And then as we started to get more oxygen availability on Earth, that's when you could have the beginnings of like the electron transport chain developing. Because even like bacteria, the prokaryotes, they still do their electron transport chain. But remember, they do it in their cell membrane because they don't have mitochondria. Okay. Any questions? 7.5? All right.